Yesterday we started talking about the nutrients that microbes used, and we mentioned a few of them, just kind of a wrap-up, a reminder. We discussed that, and I want to remind you that carbon is, this is a little tricky thing. So when we talk about life, we talk about organic life, right? When we talk about organic life, we talk about organic carbon. And I just listened to a podcast this morning, and they were discussing possible life in space, and it becomes a philosophical question. We judge life from Earth-centric standpoint. We look for carbon-based life. What if life? What is life? What if it's not carbon-based? It becomes actually a pretty interesting question here. Anyway, the life as we know it here on Earth is based on organic carbon, which can be derived by autotrophs from carbon dioxide or bicarbonate, you know. So that's inorganic source of carbon. Oxygen, that is critically important, right? Oxygen, that is critically important, can be derived from, it's kind of slow today, air mostly, hydrogen from water, okay, and nitrogen, we talked a little bit about the cycle of nitrogen, right? In fact, if you would look at it, every nutrient, I want you to be able to see the bigger picture here. Every nutrient, every chemical element that is in this table is in the cycle. Think about carbon. It exists in the organic form, like in the form of carbon in the carbohydrates or proteins or lipids. Once living things die, what happens to them? They decompose. What happens to carbon during the decomposition? What it turns into? There are two options. Which gas? Yeah, carbon dioxide. Does that make sense? And then carbon dioxide is fixed by plants, any photosynthetic organisms, to become organic carbon again. And that cycle continues. The only way to lose carbon from that cycle is if it gets buried and becomes coal or oil. Okay? Oxygen. You have oxygen in the air. It is used in oxidative respiration, becomes carbon dioxide in water, and then photosynthetic organisms, the oxygenic, in the process of oxygenic photosynthesis, produce oxygen again. Does that make sense? Nitrogen, we discussed the nitrogen cycle. Hydrogen is not, I mean, you can make a case for nitrogen cycle as well. It's not a problem. Hydrogen is a part of water molecule. Some microorganisms can produce hydrogen, mostly anaerobic microorganisms, in a process of, for instance, anoxygenic photosynthesis or different types of anaerobic metabolism. Phosphorus is a critical element that is required in several, well, three, major classes of biological macromolecules. Where do you need phosphorus? In which molecule? Lipids, phospholipids. We have phospholipids elements of the membrane. Where else? Proteins. Phosphorus is a part of phosphorylation process. It modifies proteins. But it comprises like a third of another molecule. Come on. There's only one class that's left. Carbohydrates, well, two classes then. Sorry, my bad. Huh? No, come on. Oh, what? Nucleic acids, right, because you have sugar phosphate backbone, right? Phosphate residue, that's where phosphorus goes. Okay? So, sulfur. Sulfur also establishes the cycle. I wanted to give you kind of a peek into it. So if we will start with this, it's a sulfate ion. Does anyone remember in which metabolic process sulfate ion is used? We just talked about it a couple of days ago, huh? Actually, yes, anaerobic respiration. So it serves as what? Final electron acceptor. So if it accepts electrons, is it reduced or oxidized? It is reduced. Yeah, it is reduced. Actually, sulfur is reduced. And sulfate, in the process of anaerobic respiration, can produce sulfur, like elementary sulfur. Have you seen elementary sulfur? Really cute. It's like bright yellow. Really, and crystal sulfur is just wonderful. Or can produce hydrogen sulfide. So both sulfur and especially hydrogen sulfide, they contain a lot of what we call high energy electrons, right? And these high energy electrons can then be used by different chemotrophs, okay, chemoautotrophs especially, okay, as the source of high energy electrons for respiration, okay? Not glucose, no. They're going to be the source of high energy electrons. So they're going to be further oxidized. And you've got your cycle. Does that make sense? So sulfur also cycles between the completely oxidized and reduced forms. And if you look at practically any element, we talk about, you know, completely oxidized and reduced forms. That makes sense? Right? Okay. So before we move on to metals, what you have to know about this non-metal elements, where are you going to find them? You know, where are you going to find phosphorus? Where are you going to find sulfur? By the way, in which biological macromolecule are you going to find sulfur? That's a little bit harder. It's a part of a side chain of what? We talk side chain, we talk amino acids, yes. Methionine and cysteine. That's where you're going to find sulfur. Does that make sense? Now, metals. Potassium, sodium, calcium, especially potassium and sodium especially maintain ionic strength. Does that make sense? They maintain osmolality, they maintain ionic strength. Remember we talked about the microbes that can survive in salty environments? That's your sodium, okay? Potassium goes the same way. Same goes for chloride. And the sources of all metals, by the way, are mineral deposits. Calcium can serve as a cofactor for enzymes, okay, and it's also an important regulatory molecule inside of the cells. Magnesium is a cofactor for a number of enzymes. For instance, the enzyme that replicates DNA molecule, DNA polymerase, uses magnesium as a cofactor. If there is no magnesium in the reaction mixture, there's going to be no DNA copying. Iron. I don't even know how to highlight more the importance of that metal, because some microbiologists at this point, they recite a verse by Rudyard Kipling. It's called Iron the Cold. Iron is master of them all. Practically all microbes have their own way to snatch the iron from the environment. It's a critical ion for a number of enzymatic processes in the cell, and it's kind of in a deficit in the environment. So when microbes get into the human body, they start to produce proteins so-called siderophores that can snatch the iron from the humans. Well, believe me, it's not that easy, because when we get sick, what is one of the symptoms, or should I say signs of the disease? Fever, elevated temperature, and that elevated temperature is actually what restricts the iron availability. Turns out the iron becomes less available for microbes. It is bound better to transferrin. And what's that second? Transferrin is the one that shuffles iron. I forgot the name of that protein in the liver that stores iron. Hemosiderin, okay? It stores iron. So it's pretty much like a tug of war. Microbes produce proteins that bind iron. Humans already have, well, we produce proteins that also bind iron. Whichever protein binds it better wins. If microbe wins, it's bad for us. If we win, it's good for us. It's an arms race. Does that make sense? And there are a lot of microelements that are frequently required for the microbial growth. All of them participate in various enzymatic processes in the cell. Copper often serves as a cofactor for respiratory chain. Zinc is the cofactor for DNA binding proteins. Manganese and molybdenum are cofactors for enzymes participating in oxidation and reduction. You don't have to know which one's which, okay? I want you to understand the sources of the nutrients, okay? So if I will ask you what is the major source of nitrogen, and will suggest to pick water, air, and mineral deposits, it's going to be water. Sorry, it's going to be air. Does that make sense? Because water doesn't contain nitrogen, mineral deposits are not that rich in it. Does that make sense? And also I want you to understand in which biological molecules we're going to find these guys. So if I ask which molecule hugely depends on the sulfur, you say it's protein. Does that make sense? Okay. Now, I don't remember if we talked about essential nutrient or growth factor, so I will mention it now. You just have to understand that essential nutrient is a chemical that cannot be produced by an organism itself and has to be acquired from the environment. Some microbes have less essential nutrients than others. Okay. Humans, for instance, there are 10 amino acids that we cannot make. We call them essential amino acids. We have to acquire them from the environment. There are vitamins that we cannot make. We have to acquire them from the environment. And if we don't, we get sick. Same goes for microbes. If there is no essential nutrient. Neisseria meningitidis, for instance, great example. It's a causative agent of meningococcal infection. It's a fastidious microbe that grows on the blood agar because it requires iron. Iron becomes an essential nutrient for Neisseria. You deprive Neisseria of iron, it doesn't grow. Does that make sense? That's an example of essential nutrient or a growth factor. So how microbes, how do they transport those nutrients? Again, it's kind of an arch. We're going to go back to some of those concepts over and over again. I'm not going to spend a huge amount of time on diffusion, just to remind you. First of all, the diffusion is the movement of chemicals from which concentration to which concentration. From high concentration to low, following the concentration gradient. Diffusion can happen directly through the membrane. We call it this diffusion simple, right? Which molecules can do that? That's lipid soluble, correct. Okay. Okay, simple diffusion. I should say diffusion here. Now, what if nutrients are polar, like sugars, amino acids, or it's ions? What's going to be used? What type of diffusion? Facilitated, because it is facilitated by the carriers or channels. Does it require energy? No. If transport requires energy, we call it active transport, and it goes from what concentration? Low to high. And active transport, this is just an example. I don't really care about the mechanism of sodium-potassium pump. What do you have to know about active transport? That it consumes energy, okay? And that active transport in the form of a pump shuffles small molecules. So we talk about ions, maybe small organic molecules like glucose or amino acids. Okay? What if you need to bring something large into the cell, say a protein or a food particle? When amoeba feeds, it brings in large food particles. It's not a channel for sure. In this case, cell uses endocytosis. Endo means inside, right? So endocytosis can be divided in three major classes. Phagocytosis. Phagocytosis is somewhat specific. What do I mean by that? Yes, that's a great comparison. So when you play Pac-Man, you know things that you're supposed to eat and things that you're not supposed to eat. As far as I understand, as last time I played it, I was in high school. So there are like big classes. It's like food. So you go to the restaurant and you know that you're not supposed to munch on silverware, right? You're supposed to eat food. It doesn't mean that you like all the food that's there, but you definitely know the difference. So in phagocytosis, when cells feed, they can, to some extent, distinguish between something that they should phagocytose and something that they shouldn't. It's not extremely specific. Does that make sense? So amoeba, for instance, will feed on bacteria. So it will recognize all bacteria. Does that make sense? Cell will form so-called pseudopodia. Pseudopodia usually covered with the special proteins that will recognize the food particle. It will form phagosome, and then phagosome deep in the cell. It will fuse. It's not shown in the picture. Phagosome will fuse with a vesicle known as lysosome. The content of the phagolysosome will be destroyed. Whatever cell needs will be used by the cell. Whatever it doesn't need will be thrown away. Does that idea make sense? Generally speaking, bacteria do not do endocytosis. There are some rare examples of it, but generally they don't do endocytosis. Why? Bacteria have something that isn't just easily bendable and, in the envelope. What do bacteria have that eukaryotes like amoeba don't? Which part of the envelope is present in most bacterial cells, but not present in, let's say, protozoa? Exactly. Cell wall is pretty rigid structure. So you cannot just constantly, make phagosomes bending cell wall inwards. Does that make sense? There are, like, there were some sensational examples of what looks like endocytosis in bacteria, but it's huge exceptions. So we can think, no, there's no endocytosis. Second type, pinocytosis. It's another largely non-specific process. Certain chemicals, molecules, can bind to the receptors on the surface of the cell and stimulate the formation of so-called pinosome. So pinosome, it's like cell is gulping the whatever's outside. So it will acquire whatever is outside. Does that make sense? Just drinking from the environment. Whatever's in the environment is going to get into the cell. And the third type is receptor-mediated endocytosis. This process is very specific. Certain molecules, when they bind to the receptor, initiate a series of reactions in the cell that lead to the formation of endosome, the vesicle. Okay? Whatever is bound to those receptors, this green dots, they represent proteins that react with the purple receptors. So these proteins are going to be endocytosed, they're going to get acquired in the endosome, and then they're going to be destroyed in the cell. So this process is highly specific because those are receptors. These purple bottles here, these are receptors that interact specifically with these greenish proteins. The infectious agents that use this strategy to get into the cell are viruses. A lot of them use receptor-mediated endocytosis to get into the cell. Remember we talked about viruses in the beginning, and I mentioned that they use receptors to interact with the cell. They bind to the receptor and the cell starts to endocytose them. Does that make sense? So before we move on, active transport, direction, and use of energy, endocytosis, second, endocytosis. Understand those three types, differences, very basic differences, okay? The most specific is receptor-mediated endocytosis, the least specific is pinocytosis. And finally, one thing that we didn't mention, exocytosis. What do you think it is? Yeah, expelling stuff out of the cell. Okay. Osmosis, again, to remind you, and we're going to deal with some interesting microbes today. We're going to talk about osmosis a little. So water diffuses through semipermeable membrane towards which concentration of solute? High. Water tries to dissolve a solute, right? Can't explain it here. So if you have a membrane, semipermeable membrane, and you have solution 1 and solution 2, if concentration of solutes in the solution 1 compared to the solution 2 is high, solution 1 is what? What tonic? Hypertonic compared to the solution 2. If it's lower, hypo. If it's equal, isotonic. Those concepts, hypertonic, hypotonic, isotonic, those concepts absolute. So if I tell you I have 25%, no, 25 is too much, 5% sodium chloride solution, is it hypertonic, hypotonic, or isotonic? What if it's, I have, like, hypothetically, this is not, but this is 5% sodium chloride. Is it hypertonic, hypotonic, or isotonic? Say again? Come on, develop it. You have to compare it to something, right? It can be hypertonic in relation to something else. So if I put in, if it's 5%, if I put in the hypothetical organism that has 10% sodium chloride inside, then it's hypotonic. If something has lower concentration of sodium chloride and is inside, then it's hypertonic. Does that make sense? It is not an absolute parameter. You compare to things always. Lower, higher, it is a relative term. Does that make sense? Most microbes, as we mentioned, they exist in the hypotonic solutions. So in the hypotonic solution, water will move into which direction? Where? Inside of the cells. The cell will swell. There are two strategies cells can deal with that. How do most bacteria deal with that? What do they have that prevent them from? They have cell wall. How do protozoa that do not have cell wall deal with that? Exactly. This is called a water vacuole. For some reason I think there's a fancier name for that vacuole, but it doesn't really matter. So water vacuole, they accumulate water in the organelle and then pump it out. So if you would look at the behavior of the amoeba, what does it do always? Except for pumping water. It eats all the time, because maintenance of the ionic balance in the cell requires enormous amount of energy. Does that make sense? Now, what happens with the cell in the hypertonic environment? It shrivels, it shrinks, right? Because water leaves it. And without water, cells cannot survive. Does that make sense? So how can cell deal? I deliberately don't say with hypertonic environment. With hypertonic environment, it can't deal really. How cells deal with high concentration of solutes in the environment. How they can survive. Seawater. How can they survive brine? Some can. What they do? Exactly. They have high concentration of salt inside. Some of them have this high concentration on a regular basis. So if you put them in the low osmolality environment, they're going to die. Some, like Staphylococcus aureus, can change the concentration of salt in the cell. You noticed we could grow Staph aureus on the plate, regular TSA, right? Today we're going to put same Staph aureus on the plate which contains 5% sodium chloride. So high salt. Believe me, it's going to grow because it will regulate the concentration. Questions? So osmosis, understand the movement. I'm going to ask you. It's a fundamental concept. Fundamental. It's like gravity. It's like knowing that if you drop something, it's going to fall down, not up. Okay? Direction. Where does it go? Concept of hypotonic, hypertonic, isotonic, and its relativity. Okay? And the mechanisms that allow cells to survive in hypotonic environments, both bacterial and protozoans, and how cells adapt to environments with a high osmolality. We good? Any questions? Clear? Let's move on. This is my favorite slide. I have made too many favorite slides probably, but I really love it. Because the first statement, very proud of it. Microbes live in shared habitats. It sounds very scientific, and it's very simple. It means that whatever you sample, you rarely find a single microbial species. There are going to be many. And when there are many different species, they interact. Do you agree with me? Interaction can be of different sorts, right? Can it be beneficial? Sure enough. Can it be for both? For one? Yeah. Can it be not beneficial? Neutral, right? It can be harmful for one of them. So based on those relationships, these interactions, they gain different name. How do we call the existence of organisms when they have interactions mandatory for them? Symbiosis. That's important. I'm not saying it's a beneficial interaction. I'm saying that this interaction is mandatory. You get rid of one, you get rid of another. One of the great examples of mandatory relationships is the story of liver fluke in Iceland. Have you heard about the organism called liver fluke? It's a worm, size, well, centimeter for sure, maybe a little bit more. It infects humans as well, okay? It gets in the liver, damages the liver, really not a pleasant thing. You can sometimes see it crawling through the bile ducts. Anyway, liver fluke, fasciola hepatica, you don't have to write it down, it's an example. Fasciola hepatica, its intermediate host from which humans get it is sheep. There is a European country where sheep herding, sheep production, sheep growing is one of the major national agricultural things. It's Ireland. In Ireland, sheep is like a national animal, okay? Guess what? They have about 20-25% population once in a lifetime experiencing liver fluke, just because the host is there. It used to be a huge problem in Iceland. Have you heard about Iceland? It's a small country, the population of 200,000 people, so they're pretty much all related, but that's not the point. Seriously, they're practically all related, so they really like when people from Iceland marry people from other countries. But anyway, they used to have liver fluke being a big problem, okay? And they got rid of it completely. How? Sheep was a big deal in Iceland too. So it's Iceland. Where is it located? You know? It's an island. It's an island. It's an isolated island. And they have this problem, this parasite, liver fluke, which must have sheep to reproduce. Huh? Oh yeah, they slaughtered every sheep on the island. Every sheep. Another intermediate host was the dog. Well, guess what? They slaughtered all the dogs. The problem was solved. So they get rid of one component of mandatory relationship. Second component is gone as well. Does that make sense? That's what I mean mandatory. Right? So when interaction is beneficial, so let's write down symbiosis. Symbiosis. Two organisms live together in close partnership, which is necessary. When this relationship is mutually beneficial, we call this relationship mutualism. Right? For instance, you may have a situation when you have two microbes. Let's go back to microbial communities. You have two microbes. Microbe A produces essential nutrient for microbe B. microbe B produces essential nutrient for microbe A. Does that make sense? If you destroy one, another one will die. Okay? Now, commensal or commensalism. The commensal microbe receives benefits, and another microbe doesn't really get hurt or anything, doesn't really benefit. Nothing happens to it. Often the existence of microbes, coexistence of microbes in a human gut with the human itself is described as commensal relationships. Do you understand that? Does that make sense to you? Okay? It means that microbes in the gut, they reap all the benefits from being inside a human. Food source, protection, and so on. And humans don't really benefit, but they neither get harmed. Now the whole concept has actually changed because it looks like microbes are really good for us. Some of them produce vitamins. I love that example. Some of them allow us to digest certain nutrients. Do you love sushi? Sushi, you know, this blackish thing that they wrap, well, rolls. They wrap rolls in what is that? Seaweed? Well, we can digest seaweed because of the special species of microbe that lives in human guts. We do not have enzymes. Microbe has enzymes. It breaks seaweed into the chemical intermediates that we can digest further. But initially, we don't have the meaning to deal with that. Does that make sense? So again, commensalism, one reaps the benefits, one is not getting harmed or whatever. One of the types, more specific types of commensalism is satellitism. You have a satellite microbe shown in this picture, Haemophilus influenza colonies. They use nutrient produced by Staphylococcus aureus, okay, for its own growth. So you can imagine, you can remove Haemophilus and Staph aureus growth will not be altered. But if you remove Staph aureus, then Haemophilus will not be able to grow because there is no essential nutrient. Does that make sense? Finally, parasitic relationships is when one microbe is harmed and another benefits from it. You can apply it to the relationship between the microbe and the big multicellular organism. Essentially, all pathogens that enter humans are parasites. Because humans are being harmed and pathogens replicate, they use human metabolism, human cells for the replication, human tissues. Does that make sense? Okay, that's parasitic relationship. So what we were talking about is symbiotic relationship, mandatory relationship. Can interactions between microbes be non-mandatory? And the answer is yes. There are two major types of non-symbiotic relationships. One of them is synergism. So you have two species, species A and species B. Both these species can reproduce independently from each other. But when they grow in the community, they benefit. Their reproduction rate increases. They get better protection. Does that make sense? So they experience synergistic growth. They help each other. They can live without each other, but together they feel better. Does that make sense? Now, antagonism is when two species fiercely compete, two or more, obviously, fiercely compete for the environmental resources, for instance. Not only by, just by access to these resources, but also by producing chemicals. You can see that there are bacteria here in this picture that replicate on the lawn of Micrococcus luteus, killing Micrococcus luteus, and it is killing it because it produces some sort of antibiotic. And in fact, antibiotics is not something that humans developed. Microbes have been doing chemical warfare for thousands, if not millions of years. They were fighting each other by producing chemicals. Does that make sense? We can figure out what those chemicals are and use them against pathogenic bacteria. Examples: penicillin is the essentially chemical warfare used by fungi against bacteria. Bacitracin, chemical warfare used by bacillus bacteria against everything else. And neomycin, kanamycin, ampicillin, all those microbes, all those chemicals were initially isolated from microbes as their own chemical warfare. Does that make sense? What you have to know about this: mandatory versus non-mandatory, and general definition of this symbiosis, right? And then 6 different types of relationships: mutualism, commensalism, parasitism, satellitism, antagonism, and synergism. If I give you a description like the one I gave you a couple of minutes ago, you have to recognize which relationship I'm talking about. Does that make sense? Any questions? We can move on. Good. I feel bad often that we have just this much information regarding this critically important feature of microbes. When you walk into the river, there's like Chagrin River, you step on the rocks, even if the water is really, really clean. How does it feel when you step on the rocks? It's slippery. Not only because of water, they're kind of slimy. The slime is the biofilm. The biofilm that you see on the rocks is pretty thick. It's probably comprised of bacteria, possibly unicellular algae, very likely some protozoan species, many different organisms. Of course, the liquid cultures that, for instance, right now are standing there on the bench, those liquid cultures, microbes grow in suspension. The suspension is called planktonic form, like plankton in a sea. Okay, biofilms are far more common in the environment than planktonic growth. Why it is so important? Because it's a community, and it kind of resonates with the relationships that we just discussed 5 minutes ago. So how is the biofilm formed? Planktonic cells have to reach sufficient enough concentration to start attaching to the surface. Okay, and this surface has to provide enough nutrients for the microbes to grow on. Does that make sense? So it should have some organic compounds so microbes can use it as the food source. Once they are attached, they start to produce chemicals called extracellular polymeric substances. Essentially, those are mostly polysaccharides, like part of the glycocalyx, extracellular matrix for the biofilm, sort of. Extracellular polymeric substances, or EPS, also contain proteins, contain lipids, but mostly polysaccharides. The role of extracellular polymeric substances is first to improve attachment to the surface. They work like a glue. And second, also really important role is to protect cells in the biofilm against desiccation. Does that make sense? So increases attachment and protects against desiccation. Because if you dry something, now, it all sounds very fundamental and maybe distant from your day-to-day experiences. Jerky, raisins. What's in common between jerky and raisins? Yes, they dried, right? So when they dried, microbes can't really grow on the dry stuff. They need a little bit of moisture. Does that make sense? So drying is a great way to prevent bacterial growth. EPS protects against this. Now, these microbes that settle first are called primary colonizers. So primary colonizers, once they establish certain level of communication between them, secondary colonizers are getting attracted, and biofilm starts to grow. Secondary colonizers attached to EPS contribute to EPS again. And interesting thing is that biofilms are collaborative. It's not just many species growing independently. Often some species utilize metabolites of others. Some may produce factors, contributing to growth of others, and all kinds of collaboration. So essentially, biofilm is a great example of mutualistic relationships. It may not be necessarily that all species are in the mutualistic relationships, but you're going to find a lot of mutualism in there. Okay? At a certain point, though, you can imagine the biofilm becomes crowded. Can you imagine that? Too many cells. When cells reach certain density, they start to produce molecules that are called quorum sensing. So quorum, as you may have heard, is a certain number. Like when people vote on something, you got to have a quorum. Okay? We have 27 people in the class. If we vote on when to have the exam, we want all 27 people to chip in. If I base my decision on the votes of 3 people, it's not a quorum, right? It's not enough proportion of the class. So quorum sensing is the capability of cells in the biofilm to regulate the density of the biofilm. The quorum sensing molecules are different for gram-positive and gram-negative. Gram-negative microbes use homoserine lactone, shown here in the left bottom corner, second molecule. This is homoserine lactone. Gram-positive microbes, they usually use different types of short peptides for signaling. So cells produce the short peptides and signal to other cells. It will either inhibit the growth or change the metabolic processes in the cell, making it maybe going into slowing down cellular metabolism and so on and so forth. Does that make sense? So it all works through the interaction of small peptides with the receptors or uptake of small peptides or homoserine lactone, quorum sensing molecules, into the cell. And then these small molecules, once they are inside of the cell, they will alter gene expression. Now, why? It's rather a speculative question, but what can be reason that gram-negative microbes use one type of quorum sensing molecules and gram-positive microbes use completely chemically different type? Think about the difference between gram-positive and gram-negative. What's the striking difference between them? So you're on the right track. Just make it more general. The statement, make it more general. What's the big difference between gram-positive and gram-negative? The structure of the envelope, right? That's it. So gram-positives having certain structure, they have mechanisms to transport polypeptides. Gram-negatives don't have that mechanism and vice versa. That's the hypothesis now that is being tested. Does that make sense to you? So difference in the structure of outer envelope, they determine ability to take in certain chemical types, molecules. Good? Are we clear on that? These molecules also have an interesting feature. They are autoinducers, which means that they can signal to produce themselves. They induce themselves. Does that make sense what I just said? So cell takes in, say, homoserine lactone, which stimulates production of homoserine lactone, which further stimulates production of homoserine lactone. Does that make sense? So that restricts the growth of cells in a biofilm. Now, another interesting thing in the biofilm is that biofilm, when cells have this relationships, these quorum sensing molecules may lead to the production of the virulence factors. Imagine there's a biofilm on the cell phone. Let's, for a minute, make bacteria anthropomorphic. Okay? So cells become really, really dense here. They produce quorum sensing molecules. There are two strategies for them to survive. One is to slow down the metabolism, and strategy two is to escape to other surfaces. Does that make sense? So part of the regulation can be to produce virulence factor to overcome the immunity of the host and start to escape to other sites in the organism. Does that make sense? Clinical relevance of biofilms. Why am I talking and you're not talking? Give me clinical relevance of the biofilms. Any ideas? On the joints. Excellent. Where? Mostly. Nice, smooth surface. Not that smooth. But yeah, gingiva, not that smooth. What's the smooth surface in the mouth? Yeah, teeth. So if you look at the teeth, there is a biofilm. When you brush your teeth, what do? Oh, good luck with that. You're not removing it. You disturb. It's this thing. If you autoclave your mouth, then yes, you're going to remove it. Or you gargle with like chlorine bleach for 45 minutes, you may remove it eventually. When you brush, you disturb it. Does that make sense? When you floss, you disturb it. Mechanical disturbance plays a huge role in, so essentially what you do, they just build nice cozy environment, and you come in with a toothbrush and you destroy that nice cozy environment. So they have to build it again, and then you destroy it again and again and again. Every time you put mechanical disturbance there, you essentially disturb the biofilm on your mouth, on your teeth. Biofilm growth can be found on the walls of the intestine. Most of the microbes in the intestine live in the biofilm. Prosthetic joints, catheters, any tubing that's inserted into the human body is the potential surface for the biofilm growth. And the main problem with the biofilms, it's really hard to get rid of them. Extremely hard. Does that make sense? They're really resistant. So, what you have to know, as usual, what is EPS, extracellular polymeric substance? What is the function? What is it made of? What are quorum sensing molecules? Two different types. Homoserine lactone for gram negatives, small peptides for gram positives. What does it mean, the autoinducer? Okay. Does that make sense? And a difference between planktonic and biofilm forms, but it's pretty obvious. Few more specific terms, saprobes and parasites. So both of them, saprobes and parasites, are chemoheterotrophs. Essentially, if you think about this with saprobes, unless you eat like living things, like bugs that run, you just get them and just munch on them. I don't do that, but if you do, then you're not truly a saprobe. But when the stuff that we eat is dead, does that make sense? We eat meat that is dead, like vegetables that are not alive anymore. Okay. But strictly speaking, when we talk about saprobes in relation to the microbes, those are decomposers, fungi and bacteria. Okay. Now, saprobes can be obligate. In this case, they survive strictly on the dead organic matter. So they cannot survive on the live one. Like fungus that decomposes the dead tree trunks. Okay. So how do they eat? If you think about this, neither fungi nor bacteria can endocytose, right? They cannot move, they cannot bring food in by forming those vesicles on the surface of the cell. So what they do instead, they produce exoenzymes. Those exoenzymes destroy the organic material, the dead organic material that they grow on, and then those molecules that are produced as the result of this destruction brought into the cell by the diffusion. That's what diffusion is relevant. Now, parasites, on the other hand, are the microbes, organisms that use the metabolism of the host to its advantage. Not only using it to its advantage, they also harm the host. That's important. For instance, Staph epidermidis, the microbe that lives on the surface of the skin. Is it a parasite normally? Is it a parasite now for me? No. It does use me to its advantage. But it doesn't hurt me. If I would become immunocompromised and it would cause the wound infection in me, then it would immediately become a parasite. Okay? So parasites can be ecto and endo. Ectoparasites, like fleas or lice, the organisms that live on the body, on the surface, okay? They do not get inside of the body. Endoparasites are the ones that's inside. Any infection that affects tissues can be considered endoparasitic. Large endoparasites, multicellular parasites, the great example are the worms. Okay? Different types of flukes, nematodes, tapeworms, they're all going to be endoparasites. Does that make sense? Do you see the difference between ecto and endoparasites? Now, some parasites are intracellular. Viruses are obligate intracellular parasites. They cannot survive outside of the cell. Okay? E. coli, regular Escherichia coli, can become intracellular parasite. It doesn't have to be in the cell, but it can infect cells. It's not an obligate intracellular parasite, but can become one. Does that make sense to you? The difference between obligate and non-obligate, right? Some microbes can grow outside of the human body. If you take a large genus, the Clostridia, clostridial bacteria that belong to the genus Clostridia, like Clostridium difficile, Clostridium tetani, Clostridium botulinum, they grow in the soil. They're normally in the soil. They infect humans accidentally. They become parasites once they infect humans. But other than that, they're not obligate parasites. Do you understand? On the other hand, viruses are obligate parasites. They cannot be outside of the host. So, difference between saprobes. What is saprobes? What do they do? What are the parasites? What's the difference between ecto and endo? Understand the concept of obligate parasite. Okay? Let's take a message from this one. Now we're going to talk about the requirements. Let me check one thing. I know what I'm talking about. It's just, okay. All right. So, this, I'm not trying to scare you. This is a concept that requires the most mental effort here, really. So we're going to talk about classification of microorganisms based on the oxygen in the environment that they need to grow successfully. Okay. Now we have discussed the consequences and the evolution of life on Earth in terms of the oxygen requirements. Remember we talked about it. Initial Earth was very unwelcoming and very reducing. There was no oxygen. Seriously, no oxygen. Any oxygen that was produced in quite minute amounts and released from the Earth core, Earth crust, was immediately used to oxidize chemicals. So eventually it led to the development of anaerobic life. And the first cyanobacteria, first photosynthetic organisms, remember it wasn't plants, it was cyanobacteria. They produced oxygen as the waste that was immediately used to oxidize iron in the oceans. And there are vast amounts of iron oxide on the ocean floor. It is found, really, it is found, and it dates all the way to the time when first anaerobic prokaryotes appeared. So it is confirmed by the findings. And remember, we talked about photosynthesis, okay? So there were two things, two roles for photosynthesis. One was to make complex carbohydrates from CO2. What was the second? Photosynthesis was the way to protect cyanobacteria from what? Remember? Oh. There was no oxygen, there was no ozone layer. UV radiation, exactly. So you used the kind of buffered energy of UV radiation and directed it into the chemical synthesis. But oxygen concentration rose over time, millions of years actually. And eventually it led to the evolution of aerobic microorganisms. And aerobes, they got the evolutionary advantage. What was the evolutionary advantage of aerobic metabolism? Compared to anaerobic, it is what? More efficient, yes. So, now the world is aerobic, and forever will be until we destroy it. So let's see the 5 major classes of aerobic microorganisms based on the oxygen requirements. This are obligate aerobes, okay? So these microbes cannot grow without oxygen. Does that make sense? Cannot, absolutely. They must have oxygen. Does that make sense? So if you would take a medium in a tube, medium that semi-solid medium, okay? Semi-solid means it's not like solid like agar, it's not liquid like the one that's there in a rack. It allows microbes to move a little. Does that make sense? So aerobes will occupy space on top of the medium and will not ever go under. Why? Yeah, there's no oxygen in the bottom, right? This is obligate. B is the obligate anaerobes. On the contrary, these guys will occupy the bottom of the tube. Why? Not only they don't need oxygen, they cannot tolerate it. They're getting killed by oxygen. Does that make sense? There's a microbe called Clostridium perfringens. It is a causative agent of gas gangrene. It's obligate anaerobe. What is the treatment? It's not the only treatment, but one of the common treatments for Clostridium infection. Yes, hyperbaric chamber oxygen. Yes, it's very effective. Okay, for instance, another example. Have you ever heard about a disease called peritonitis? When the bacterial content of the intestine accidentally spills over into the abdominal cavity. Intestine is inhabited by anaerobes, and species Bacteroidetes, well, genus Bacteroidetes, they are obligate anaerobes. If you have a patient with peritonitis, and there's an infection, you open the patient up, clean. Can you clean completely? No, you cannot. I mean, you can kind of suck out the stuff, but you cannot sterilize the guts. In many cases, those patients are not stitched back right away. The abdominal cavity have to be exposed to oxygen to kill all obligate anaerobes. Otherwise, you close the wound and they start to replicate, because it's not really high oxygen in tissues, surprisingly. Okay. Does that make sense so far? Facultative anaerobes. Now these guys, first of all, don't read the description. Look at the tube. Where do you see the highest density of the microbes? Where there's oxygen. Do they grow without it? Or with the low? Yes, they can grow without it or with the low concentration of it. What does it tell you? They prefer higher oxygen, but can grow without it. Can you make a leap of logic and, thinking about metabolism, suggest which types of metabolic processes can they use? I'm giving you a hint. They can use two different types of metabolic processes. Two different ways to make ATP. Exactly. Aerobic respiration when there is what? And when there is none, they will use fermentation. Does that make sense? Does that make sense? When oxygen is not available, they will rely, well, I should cross away anaerobic respiration. Without oxygen, they will rely on fermentation. Does that make sense? Great example is Staphylococcus aureus. Now, I wanted to say something and I completely forgot, just totally forgot. Oh, the name. Why we call them facultative anaerobes? Anyone, what does word facultative mean? Facultative is opposite from obligate. Obligate means what? Must. Like, right? Must. Then facultative. Yeah, I don't have to. Okay? So facultative means that they may be anaerobes. They can be anaerobes, but they don't have to. Does that make sense? So facultative anaerobes can use fermentation when oxygen is not available. Okay? Air-tolerant anaerobes. Is there any difference in growth between aerobic and anaerobic conditions? No. So these are anaerobic microbes that can tolerate oxygen. Does that make sense? They don't use it at all. They don't use aerobic respiration. But they can grow in its presence. Good? And finally, Goldilocks, or microaerophiles. Okay? Where do they grow? Which has very specific. There we go. That's why they called Goldilocks. So Campylobacter jejuni, I think 5% of oxygen in the environment. Okay, it's just right, not more, not less. Well, I mean, if you do 5.5, it's probably going to grow, but worse. Okay, so you have to understand these 5 different types. And if I provide the description, if I tell you, Staphylococcus aureus uses aerobic respiration when oxygen is available. It switches to fermentation when it is not. What type of microbe does it belong to? Facultative anaerobe. That makes sense? So you have to, and vice versa. I give you facultative anaerobes are, or obligate anaerobes are, and you have a choice. Okay? Did I make myself clear about what you have to know here? Now, what is the reason? What are the reasons we have aerobes, anaerobes, facultative anaerobes, obligate aerobes, and so on and so forth? What are the reasons? These words. But what are they? There are certain species, well, chemicals that are produced during the respiration. Do you remember how we called them? You actually see the acronym here. What does this acronym mean? Yes, reactive oxygen species. Right? And among those reactive oxygen species, 2 seem to be most important. It's hydrogen peroxide and superoxide anion. Okay? So peroxidase is the enzyme that neutralizes hydrogen peroxide. Okay? Usually in the acidic environment. Okay? It provides substrate that is being oxidized. Okay? And essentially, hydrogen peroxide produces water. What's most important is that now hydrogen peroxide doesn't hurt the cell. Does that make sense? That's peroxidase. Catalase, right here in the bottom, leads to the intramolecular oxidation and reduction. You don't have to know that statement, intramolecular oxidation and reduction. The point is, it neutralizes hydrogen peroxide, creating water and oxygen. Okay? And superoxide dismutase neutralizes superoxide anion, which is extremely powerful and dangerous reactive oxygen species, producing hydrogen peroxide and oxygen. You can imagine that this molecule can be further neutralized to other reactions. So those 3 enzymes, you have to know them, you have to know what they do. I don't care about the reaction. I'm not going to ask you the reaction. No. Don't worry about this. But if I ask you which one neutralizes the hydrogen peroxide, it's not superoxide dismutase. It's strictly peroxidase and catalase that we talked about. Does that make sense? Was I clear on my expectations? I always want to be absolutely clear, so don't tell me you didn't tell us before. Okay. Now, let's go back to the genetic differences. What if the microbe has enzymes for all three? For all three, okay? They will probably going to be facultative anaerobes or obligate aerobes. Obligate anaerobes, if I'll go back for a second, you see where they grow, obligate anaerobes? So they cannot stand oxygen, which means they have neither of these three enzymes. They do not have means to protect themselves from reactive oxygen species. Is that clear? Good? Awesome. Aerotolerant anaerobes, remember? They can grow in the presence of oxygen. They don't use it. So reason for that, they have superoxide dismutase, which allows them to survive in the atmosphere, but they do not have enzymes to handle hydrogen peroxide that is formed in aerobic respiration. So they can choose it. Does that make sense? That sounds very biochemically fundamental, but here's the diagnostic value of it. Staphylococci, as we mentioned, are facultative anaerobes, which means they have catalase. Okay, this enzyme that facilitates this reaction. Streptococci are aerotolerant anaerobes. They do not have catalase, which means they cannot facilitate this reaction. If you take a little bit of a culture of Streptococcus versus Staphylococcus and add, on top of the culture, you add a drop of hydrogen peroxide, catalase negative microbe on the left will not produce any oxygen. Catalase positive microbe on the right will produce oxygen. Before I move on, does that make sense? If the microbe has catalase, it can neutralize hydrogen peroxide, make an oxygen, and you can see the bubbles. It's as simple as it can be, an extremely convenient method to distinguish between catalase positive and catalase negative microbes. Does that make sense? So you have to know the diagnostic value of catalase positive versus catalase negative microbes. Okay? That simple test, you have to understand how it works. Right? Now, how do we handle anaerobes? It's a big problem. You take them out from the environment that they're used to and they die. Seriously, they die right away. My wife works in the lab that studies inflammatory bowel disease, things like Crohn's disease, ulcerative colitis, and they study the role of microbiome, intestinal microbes as well, in the ulcerative colitis and Crohn's disease. I told you before that microbes in the gut are anaerobic. You open, well, they don't use humans, they use mice. You open the mouse, you take the gut, they're all dead, like 2 minutes. Really, they're dead. So they have to jump through millions of hoops. They have an anaerobic chamber to work with, these guts, to put them on the blood agar and so on and so forth. It's really pain in the neck, okay? I don't want to make a pun out of it, okay? Pain in the neck to work with anaerobes. You can use the special chamber. Looks pretty sophisticated, or the hood. So in this hood, you fill it with, say, nitrogen. Okay, so you substitute all the air with nitrogen. This chamber right here, we have several anaerobic chambers right there, and you see that cabinet, the row of cabinets on that wall, the rightmost cabinet. From my vantage point, I see at least 4 chambers. Any idea what they are? The pickle jars. We make anaerobic chambers out of them. Any idea how? We can stick a candle into it. Light a candle. So we put microbes, put a candle, light a candle, burns out all oxygen. There it goes. It's anaerobic. It's a lousy chamber, to be honest, because the lid is not sealed, so there's leakage there. We tried it last semester. It's hard to say because we didn't have any true obligate anaerobes. We tried to compare aerobic versus anaerobic growth. There were some minor differences. We can try it this year. It's fairly easy experiment. You inoculate plates and just stick them in there. I was planning to do it probably last week. If you have pets, you will need to collect the poop from pets. So some microbes also require CO2 to grow, and they are called, I'm not going to put it here, so these are called capnophiles. Okay. The Campylobacter jejuni, same Goldilocks microbe, is an example of the capnophile. Okay. That makes sense? Can we move on? I'm trying to see. 2, 3, 4, 5. Just want to do something in the end. Okay, let's talk about pH and pH requirements. Listen, that's as simple as it can be. We have 3 classes of microbes: neutrophiles, acidophiles, alkaliphiles. Okay, neutrophils grow the best at the neutral pH, acidophiles grow best at the acidic pH, alkaliphiles at the alkaline pH. Most pathogens are neutrophils. There are some acidophiles that we use on a regular basis. Okay, like yogurt, buttermilk, sauerkraut, cheese. It's all acidophilic bacteria. Okay. I think there's a species, Lactobacillus acidophilus, something like that. So they like acid. They grow in acid pretty well. They're not champions, though. There are a couple of species of archaea and bacteria. Sulfolobus, that's archaeal species. They grow at pH 3, but not only it's acidic, it's also pretty hot. It's practically boiling. Ferroplasma grows at pH 1. It's 100 times more acidic than your stomach content. It grows in the mine sewage. The copper mines, they have acidic runoff in the mines. This microbe grows in this runoff. If you put it at the normal pH, it dies. It becomes all wrinkly and dies right away. It cannot survive at normal pH. So that is quite unusual, being acidophile or alkalifile. Reason for that. So what happens? So acids, okay, they denature proteins and break down hydrogen bonds. Break down hydrogen bonds, which molecules, macromolecules, nucleic acids, and also proteins, because proteins, when they fold, they acquire the secondary and tertiary structure. This structure is largely formed by hydrogen bonds between side chains of amino acids. Okay, so they denature DNA, they denature proteins. Bases are often considered to be even more dangerous to cells, because bases inhibit the critical process in the cell that depends on hydrogen ions. They neutralize hydrogen ions and they inhibit this process. Which process in the cell hugely depends on hydrogen ions. Yes, cellular respiration, right? Because it's hydrogen transport across the membrane. If you don't have hydrogen ions, you boned. You don't have anaerobic, you don't have respiration. So that's bases. Now there are microbes that can live in the extremely basic alkaline environments. The lake natron in Tanzania, I read pH something around 10. There are archaea and bacteria that can live at this pH. Okay, that makes sense. Okay, I'm gonna give you a break.